Welcome you, sir, for this lecture. It is a homecoming to Dr. Naladevi, the alumni to this department. It is immersive under the chairmanship of Dr. Marimuthu, Professor Plant Ecology. He worked in mushroom. Then later he joined in ICR service ARS. Then he completed his PhD from Udaipur. Sir, am I right?
Yes, Rajasthan.
He has completed his PhD from Udaipur. Then the brief period he served at IARA also. Later he moved to IARA Regional Station, Wellington. So he's a very good worker and he's always having passion towards the mushroom. So always our mushroom scientist, Dr. Trimunamala, always interacting with Dr. Naladevi for the work on mushrooms. And he also serving as member for Dr. Trimunamala student. So he's a very good scientist. So we can hear this lecture on combating biotropic pathogens of wheat by exploring the host-resistant genes. It's a very important area. You all understand that biotropic pathogens is very difficult to manage. So the option left out is resistant genes. That is the durable one. So if you go for any kind of chemical management or biological management, they are not fruitful. So the option left out with the scientists are the resistant genes. So he is currently working on this area. So we can hear from Dr. Naladevi on this topic. So now I request Dr. Naladevi to deliver the lecture.
Let me thank this wonderful department. In fact, I'm looking was my very good junior in the same college, Pilipala, and here also, and all my colleagues, friends, and their students. It's a great privilege for me to be coming to this department. Of course, I am alumni for the three campuses, IRA and Udaipur, and of course our TNU. So obviously, I always like to have the impact with our own university. It's no need of introduction about me further, and already that I'm going to go. It's a journey in the sense because we are the National Agriculture Research System, and we go to work according to the mandate of the institute and the mandate of the top. And of course, that Rangapan told that the work on mushroom is my personal interest, and the patients still we are okay. And this the post resistance. I think from undergraduate we have studied the management of the crop diseases. So first the point to break that resistant varieties. But how to pick up the resistance? It's really challenging, and particularly for the some of the pathogens. Pathogens like the biotrops, where you are not able to culture the pathogen. When you don't have the pathogen, of course, host will be there, but unless you don't have the pathogen, proper pathogen or proper the race or the proper the pathotypes, we cannot identify any resistant source. So there are a lot of challenges with this biotropic pathogens. So here I want to discuss, maybe you want to already Dr. Rangapan told, very recently we made a breakthrough by whole genome sequencing of the biotropic culture. It is found, I think, for first time in India. Of course, in other countries too, few countries they have assembled the genome, but all were incomplete. For the first time we used the hybrid mode of the hybrid technology, and we could publish recently it has appeared in the journal Panjai. So with this little background, let me take you to the different aspects, different very important parts. As a pathologist, what we should keep in our mind when we are working on the challenging pathogens. So this here, you know that the post resistance is intentional use of the resistant cultivars. Plus, we are intentionally identifying the varieties to control their pathogen. without using any pesticide or other means. And the way back, Simons classified the resistance. Otherwise, as a pathology, we know only two types of resistance. One is vertical resistance, and one is horizontal resistance. It's a common one. But there are other components like the major gene non-specific resistance. Of late, we are working on the non-host resistance. That is non-specific resistance and vertical resistance. Of course, it is gene for gene hypothesis. And before 1942, we made a breakthrough that the virulence genes from the pathogens very important play a role in the vertical resistance. And of course, you have the horizontal resistance, where most of the breeders could practice horizontal resistance. And of course, the interaction resistance, the effect of the population, multiple population or the vital mixers, where in countries like India, we are growing the different varieties in different growth ecosystem. So where the interaction resistance will work. So with this background, if you take back the events, the resistance, particularly for the resistance, what happened in our country particularly, after the invention of the Boda mixture, and we started working on the post resistance in the 20th century, and we understood the genetics of resistance. In fact, that helped us to identify the origins, the resistant genes from the host. So unless we identify or understand the genetics of the genetic basis of the pathogen and the host, and we cannot identify the origins. So this happened in the 20th century. And later on, of course, already told the gene for gene model from flow, and from there we started our journey. And from the 20th century, we studied a lot of chemistry and molecular biology. I think the 90s, MSCR PhD thesis, even today, we are estimating the biochemical factors involved in the peroxidase, polyphenol oxidase, lot of genes. So we understood the chemistry and the molecular biology, both the side, and we could identify the mechanism of the resistance. Because that is very, very essential. Unless you know the mechanism of resistance, you cannot proceed further. Now, very recently, we are identifying the discovery of the specific genes which are encoding for the either it is resistance or susceptibility. Because very recently, we are taking care of these susceptible issues also. Because most of our removing varieties, very classical varieties, even the varieties which made breakthrough in the Green Revolution, as now it is no more because of the susceptibility. So that is the recessive gene is also very important. So that part we are identifying by encoding the identifying the genes. And of course, very recent model that zigzag model is the molecular events. I think most of the students very interestingly working on this molecular events of the post-flux interaction. So now we are going on this part, and the first one is the discovery of encoding genes where we landed with the problem that I will discuss with you. Of course, advantage everybody knows it is ecological, sustainable, compatible with other crop production components. Particularly in case of wheat, I am talking about wheat, because in wheat we are not giving much irrigation as compared to rice. So very less irrigation with the fertigation, because fertigation is important. So these resistant varieties are very compatible with these two components, and of course the socio-economic viability. Because in North India, if you go the Kharif season and the winter season, we have huge areas cultivated by wheat. So the farmers without any extra cost on the varieties, and we can grow without any pesticide. It's very eco-friendly. This is the three major advantages when we are working on the wheat, which are resistant to most of the viral diseases. Coming to the post plant resistance, the major components. As a pathologist, we should be very, very clear about the selection of source of genetic resistance. You can think of why we want to bother with this. This is the job of breeder. It's not like that, because the selection of source of genetic resistance is very, very important for the pathologist, not for the breeder. Because pathologists only know that the knowledge of the pathogen biology and that everyone. First point. And second point, then we will have our own field glasshouse or the controlled consent facilities. And the availability and accessibility of the germplasm and the genotypes. Of course, we need the help from the breeder. So these three components very, very crucial for us as a pathologist. And the remaining thing that the utilization of resistance, that is the job of the breeder. Because he has to think of what kind of breeding method he will follow and in which season he has to grow. So those things he will use, and the deployment of the resistance and the release of the varieties. Again, it's a combination of the pathologist and breeder work. And most of the institutes and most of the crops, we could see that the pathologist's job ends where you go to score the material, you go to say whether it is resistant or susceptible, our job is over. It's not like that. Because we should understand, and most of the things it is, that's why most of the time the conflicts arise between the breeders and pathologists. And they themselves will claim that after 5 years of release of varieties, you only give the varieties as a resistant. Now we are telling it is a susceptible. So they blame again will come to our part. Because that is only because we are not understanding the mechanism or the genetics of the host. It's very simple. It's not a we know, I think Dr. Nagarajan is a very famous epidemiologist. The plant pathologist is no more now. And he worked on the senior part. We will discuss a little bit on that. So he was a very classical breeder than the pathologist. Then only he could make the trust epidemiology very successful more. So we should understand the genetics, the basis of the host, genetic base of the host. Then only we will be able to work on the pathogen resistance. Now, over the period, what we learned from the different crops, starting from a career from sugarcane for about 5 years, then arid agriculture in Rajasthan, our desert of Rajasthan, where we worked on date farm, very tough crop, very tough crop, very tough climate. And of course, moving to the IRA, now it is a weeds. So we see that the very big research gap within the pathologists that we go to establish a very systematic protocols in procedures. Most of the time, you are following the procedure protocols, which are very age-old practices, and we are not maintaining the culture according to the pathogen. Any pathogen will maintain only on previous runs. It's not like that. If you see the pathogen biology, the hepatitis require different kind of nutrients. The Artenaria require different kind of nutrients. Even within the Artenaria brassicae and brassicicola, both are same, but it will need it's a little bit of change in the amino acids micronutrients to maintain its viability. So these are the very crucial point. What we learned from our own experience, I could suggest to that we should have a well-established protocol, and of course the maintaining the facilities. Minimum facilities in pathology, at least one deep research for the student, and the proper minimum facilities to prepare the culture and maintain the cultures, and pass on the cultures to the next students or the next staff. So these things we want to maintain, particularly for the biotrophs. I am talking about the biotrophs, and we are not bothering about the necrotrophs, because once you use the biotrophs, once you use the host, you will not find the pathogen. Unless the host is there, you will not have the pathogen. So we have to maintain both side by side, host as well as the pathogen. I will take you to how we are maintaining the biotrop culture in the host. So we have to maintain both these things. Now, what are the reasons why we want to prepare the resistance, host resistance and wheat? Because we have a huge number of pesticide companies, huge area for a lot of multinational companies. Suppose if the stem rust occurs or leaf rust occurs, you can go for the large scale. Even now the drones has come, you can go for the spray. But it's not like that, because the diversity within the genotypes, wheat is one of the unique, very unique and very tough crop to work. If the whole genome is very big size, almost acquire with the human genomes. They took a lot of time to sequence the whole genome of the wheat. It is a better plant, and the repeated use of the existing organs. In India, we have about more than 500 released varieties of wheat. But of late, most of the varieties, or even 50% of the varieties, are susceptible to either leaf rust or stem rust or cautionary or another. How it has happened? Because we release in our wheat improvement program or protection program, we have very stringent protocol. Unless the host is the variety is resistant, we will never allow for the release in the national state. But we could see that most of the varieties are become susceptible because we are using the same genes, same kind of cascade of genes for the developing the resistant varieties through breeding program. And of course, they change in the disease pattern. The first part you can see, I think even my undergraduate, I studied that wheat means first you start with stem rust, Vaccina gramineaceae disease, then leaf rust, then yellow rust, head scab, then powdery mildew was a minor disease. Even I didn't bad about the concept. But now you changing this graphic pattern, you can see the yellow rust stands first. It is a very devastating disease in the North Indian graphic pattern compared to other diseases. And there is a big vigilant theme on only on this yellow rust in North India. And of course, leaf rust, stem rust. Now just the powdery mildew has come up due to maybe the climate change or the varietal change in the pattern. Of course, the head scab caused by Fusarium gramineae, seed-borne fungi, a very important pathogen compared to the carnal. Carnal now we are able to minimize due to the change in the cultivation practice of these. And of course, the powdery mildew, so you know very well that it will produce a lot of the asexual chlamydia and then of course sexual things. And this slides already I shared some time back. The same slides, you can see the symptoms and the leaf rust, stem rust, and of course the yellow rust. Now that's what I told the Dr. Nagarajan's contribution is the Vaccina path. Why the Wellington station is important for this, not only for our country, the international level, because only in Wellington only you will have the wheat crop and the pathogen growth the year, no and etc. It's a very interesting location. Even as a pathologist, I myself didn't know that this much of importance when I was serving other spaces. So any time you come to Wellington, you will find the wheat as well as the rust. That is the beauty of the station. And because of that, and Dr. Nagarajan initiated the work on the Vaccina plots. And this is a general principle. In plains, normally the wheat is cultivated in the rabi season, winter season, and it will be harvested in the month of April, May. Now you can see the temperature more than 50, 48, 50 in the North Indian region. So this time you won't find any inoculum. But how the infection takes in the next year, that was the issue. So they worked the case in method started work on this because when the wheat is not there in the plain, we will have case. That time the rust inoculum is there. Now the southwest monsoon will start June, July. The cloud will carry all the windows to the plain, Peninsula zone and central India, where we have the infection. That is the path, Vaccina path that will be a simple way we worked out. That time the wheat cultivation was there in the lower funding hills and even in the Middle. Now it is not there because the farmers shifted to cultivation of carrot and other commercial crops. Wheat is no more cultivated, but our station being a research station, we have a wheat plant and therefore the inoculum still it is there. Because this wheat is very effective system because you know that the management of biotropic is very challenging. Without fungicide, mass production of the iridospore or any other spore from the biotrophs, and every of the spores we can spread a very long distance and rapid evolution of new pathotypes. Because in wheat there are 3 species. Triticum yeastivum, it's called bread wheat or the chapati wheat. What we are making chapati, it's a Triticum yeastivum type. And Triticum dipacum, the Hawaii, the Sambaku. Upma that is the Triticum dipacum. And then Triticum durum, where you have the noodles and pasta and all this. So otherwise we will think that the wheat is only one crop, only one species. It's not like that. These 3 species, the same genus. So 3 species, because of 3 species, you can, I already told that the genome is very big size. Then you can imagine that how much the host diversity will be there. Then how the pathogen will act to accommodate itself in all the 3 species. Now you can imagine that how many races or how many pathotypes will be developing throughout the season or throughout the country. So that is the beauty of this both rust pathogen and also the 3 species of the Triticum. So therefore, we are having a lot of pathogens, new races. And so far in our the national repository at Shimla, we are able to maintain 145 races. That is standard characterized races. There are so many unknown races also. So therefore, the government made a recommendation that the rust-free material only will be used in this country. But there will be always a threat because it is a ureoscope, it will carry through the wind. It may travel to one country to another country, you cannot restrict it. And of course, our movement also. We are moving internationally. As a pathologist or as a breeder, as a human being, we are moving to country to country and we will carry the norm. That you cannot avoid. So these are the two risks we are having. And already I told that while in the Indian context, the resistance breeding or the resistance source identification somewhere in 1920s they initiated. But 1960s a systematic work started in India. They initiated with the TPM, that is trap plot nurseries. Still we are having this nursery throughout the country. And of course, later on we included the material from the South countries. Because neighboring nations we are growing this material and we included that. And I already told that in the Indian Institute of Wheat and Barley, we have the national repository. So all the any new race or new pathotypes we have to submit there. You cannot take like any other culture without any proper procedure or proper permission. In fact, we used to, as a pathologist, we used to tell it's illegal collection of the culture. That is a punishable offense in the system. I cannot take any new race from central India and work without the prior protocol. So because it will, anytime it may take outbreak and wheat production will be affected. So all these collective efforts, and so far I told that the Indian the wheat resistant lines, we have about for the stem rust we have about 22 genes in the post we have characterized in Indian wheat varieties. Similarly, for the leaf rust 19 genes and the yellow rust only 6 genes because we already told the yellow rust is coming up now only. These are the total number of genes we identified in our own Indian wheat varieties. Any variety if you take, at least 1 or 2 genes will be there. And this in our station's activity. We are working on 2 types of resistance. One is the seedling resistance, or all the all kind of resistance, and the adult plant resistance. For us, the seedling resistance is very important because we used to get a lot of material from the breeders throughout the country. All the breeders or wheat breeders they used to send. This is mandatory for us, and we want to test all these materials in the seedling stage. Then, of course, any material they want to have for the adult plant stage, then we will test the adult plant screening. So far we have identified few adult plant resistant genes like LR34, 46, LR means leaf rust. Like it is for stem rust means it will be SR. If it is for yellow rust means it will be OR genes. So these the 3 category of genes will be there. And in our case, particularly in Wellington, either it is for overwintering or it is for rust or any other disease. This XR reaction is very important. Hypersensitive reaction or hypersensitive response. Because in the course we studied that hypersensitive reaction, that's one or 2 lines. But this is very critical part where the other kind of resistance mechanism will start. Unless any pathogen interaction fail in the XR reaction, then it will not open any kind of resistance. So the hypersensitive reaction we will look into very critical. Suppose any pathologist or any technician by oversight, if we miss his resistance, then we will be missing the reaction also. Thus in the field it will express the symptoms. So it will be very critical, and we are using this mechanism to knock out any number of genes. Even any breeder may send 500 or 1000 germplasm or the F1, F2, F3 generations. So we will be looking for this kind of resistance, and this is the reaction pattern. Different genotypes, you can see each genotype will have its own reaction pattern. This is for the leaf rust. You can see only some generates the hypersensitive reaction. So this kind of reaction will estimate and will shortlist the resistant source. And of course, this is the methodology. After 7 days of the seedling, we used to take the immunospores and we inoculate and incubate for 48 hours in the chamber. Then we'll take it out and we'll keep it in the glass house. And we used to have the 0 to 4 scale, it's already established. And suppose if we want to analyze is there any new race or new pathogen, then we go to follow the technary value system that was developed by Nagaraj and group. So still we are looking using that scale. It's very classical, very interesting scale. If anybody is interested, I will discuss it separately later on. So we close there. And based on the technary value, we can designate whether it is a new pathogen or new race. It's a separate procedure is there. And this, of course, the reaction pattern, normally we used to follow. Already I told the hypersensitive reaction. This for the stem rust. And I told that how to establish the infection. Sometimes, because now if you see in Nilgiri, so the temperature is more than 0.4 or daytime, very high temperature. But night it will be very cool. Sometimes you will get rain also. Within 24 hours, this kind of the fluctuation around the environment or weather conditions there. So obviously, the rust incidence in the field is very uncertain. But we should ensure that whatever the check line or the suspected line we are putting in between the test lines, we should get at least minimum 40 to 60% of the infection. Otherwise, we cannot be scoring. So for that, we will use the artificial cultured indoor store in the field, early in the morning or late in the evening. Then we will establish the disease. I told that different R genes present in the host. The leaf resistant genes. So far, globally, we characterized 80 leaf resistant genes in the host plant. Similarly, more than 60 to 70 PM genes, coordinated resistant genes in the wheat. Similarly, for the yellow rust and the stem rust, the number of genes, this was the number of genes for the yellow rust host plants. I told that the breakdown resistance, because since we are using too many genes, the same genes repeated the group of genes, but anytime you may expect the breakdown of resistance. I think most of the pathologists know that the boom of first theory, still it is working on the wheat. When you are having the new virulence and it's together it will spread, and when it infects the new resistant varieties, it will be a boom, because when you are cultivating any new varieties, it will be a boom for the country, for the production, but anytime it will break the resistance. And this is the powder mildew where we initiated this work about 6-7 years back. And here actually, this how we have to score it, because the same leaf is having both the 50% by powder mildew and 50% by virus. So it will be a very tough job. When the breeders send for their rust resistance, I am getting the powder mildew. You can see the adult plant completely vanished. No space for any rust to get the infection. So how to score it? So when I say that there is no rust in your plant, only powder mildew is there, the breeder won't agree. You have to establish, you have to give the proper justification for the material. Because they are hard effort about 6 to 8 years to develop a wheat variety for them. So one season, if I say that this variety is no infection in the rust, if I say the same variety, we send because our breeders are the very intelligent breeders. The same material they send in the next year also with this different code. You won't find, because as a pathologist, we never know that what is the progeny, what is the pedigree, what is the family. They have the coding and decoding system. That's good also. It's very excellent system. But very interestingly, they will send the same material for the re-evaluation the same season. So that time, if I say that this is the same material, now it is susceptible to leaf rust members. I by last time season only told that it is resistant to all the rest. Now we are telling susceptible. So because the problem starts with the pathogens. Because it due to changing the weather condition or the actual environment load, you will not have the infection. And most of the Indian varieties, for the information, most of the Indian wheat varieties are open. Because we never worked, we never thought of that this disease will become, it will take upper hand in the later stage. We ignored. So my humble suggestion to the pathologists that let us not ignore any disease which are occurring in the particular post. It may be a very minor disease. If today it may be minor, maybe tomorrow it will be the future to work on this. You can take any classical example, rice, consumate, Laxmi disease. I think in our postgraduate for the sampling we used to sit where that smart ball in the lab. But now you see this is the number one disease in the rice. So similar case, as a pathologist, let us take each everybody in the same category, whether it is minor or major, economically important or not. So this is one classical example, and we have so many origins. And we first track that the pathogen survives through the resting state, that is rest of the classification. It was not reported from Wellington because everybody was studying that when you are going for lead first scoring, we are getting the probability. And everybody asking from where the probability comes. You are not maintaining anything in the classes, you are not working on anything like that, but in the field you are getting huge infection other than the rust. So there we could see that if the pathogen survives through the resting stage in Cashmere, then the molecularly we confirmed the latest foods. And these are some of the lines or the varieties we identified over the period, last 10 to 12 years. Some of the materials we identified. Perhaps this may have a large number of PMGs, 4 into genes. What I listed 80 LR genes or 65 SR genes, 10 to 12 VR genes. Similarly, the PM genes globally they are characterized about 65 to 70 PM genes, but none of the genes have been characterized from India. Because we ignore, it is not our fault, because the disease that time was not there. Now it is taking the upper hand, but we don't have anything to develop any varieties because we don't know the any genes, what are the genes available in the host. So from where to start. So this is the big challenge for us. And of course, we with our team, we would have some of the materials and how we culture. This I wanted to discuss. How people ask you how you culture the powder and in this quiet time. Because some literatures are there of appeal for the grapes powder and other powder. But sometimes we also use our own way of thinking and way of methods. So this method it is already described for the grapes. Here we use the plain agar. It is as good as the same vertebrate with the plain agar sterile medium. And this surface sterilized beam, we put on the water agar medium and we inoculate the portability medium over them. Because the agar medium is supplemented with the benzimidazole, so it will take care of that it won't allow the leaf for a long time for the senescence. Tide over the light tissues. So it will give the support that chemical. So we put that we inoculated, and after like any other pathogen, maybe after 10 or 15 days, we could see the positive parties. So this we track and we utilize as for mass multiplication. Now okay, we got the culture, how to multiply. Again, the same natural method we tried by test tube. We put the seed and the seedlings grown. Instead of putting on the culture on the PDA strands, we put the culture on the seedlings by the aseptic culture. We kept it, and sometimes because the seed will carry a lot of it. We don't know what are the microbes are. Though we are putting the surface sterilization, so many methods, but there are so many internal seed-borne pathogens like smut and other things. Of course, the smut won't come here. But for the example, the two pathogens, one is the Physium graminearia, the other is the Artenaria, that is a block point we use here. So these pathogens, they will present in the inside the deep embryos of the seed. So if you cut the embryo, the seed will not germinate. So we put that, but we had the seedling. We can see the seedling there, but where the healthy seedlings are there, we could get the healthy colonies of. So this way we purified about 275 isolates. It was very, very tough job. Fortunately, we had a DPT funded project. The DPT was so happy, so excited to fund this project. And we got it, and we three institutes collaboration, one from Shimla and another from Karnal. We worked and we standardized this method. And now we multiplied on the post. It is all the aseptic techniques. Because we designed any pathologist, if you come to our station, I will show you the glasses and not the body hooks. Controlled condition, how we design. We made a chamber inside like your BOD incubator. It is a big chamber, but like our BOD incubator, the cells we made a chambers. For each group of isolates, we will maintain that particular chamber only. Within the chamber, we made a small micro cans made up of the polycarbonate sheets. Because we cannot use the glass into the huge weights. Polycarbonate sheet, transparent sheet, very cheap as well as very economical and very transparent also. It uses the aluminum frame, very weightless and very convenient to use with workers. So with this our own creation, our own thinking, we established and now we are able to test or screen n number of materials on the board. This is the first step with kinds in the country, of course in the global level. We have established this facilities of the region, and now we last 2-3 years, the government, all the wheat improvement program, they are sending their material for seeding resistant test building. It's a great contribution to this station and as well as the system. Because as a pathologist, we played our role in a different way, and now the breeders are very happy. At least what are the earlier the role guiding method, now they have method to test their material. And this is the reaction I told you. You see the result. We will inoculate because we got a few culture, we got one method to inoculate like this, and we got some of the material you find this. So this once you find this kind of material, you can pass to the breeder. They will be very happy to play for because they know how to multiply, how to make a combination of classes. So many things they do. And of course, the other part I told that the whole genome. Because we committed, I think at one stage, I thought that I wrongly committed to the DPH that we will be doing the whole genome sequencing problem. Because we could not get the DNA from the conidia. You know that the probability conidia is very transparent, very tiny nuclei, and that how to extract the DNA. And of course, for ITS and other things, we have used our one thing. The whole genome will be this very tough situation because we love them to that. So the average genome people 2-3 times they came to visit our station and we supported them in a different way. And they when we worked taking a lot of support from the classmate. He's a very excellent person, very brilliant guy in junior country. So we discussed many things in the behind the screen because even at one stage the company was ready to return back the money what they collected for the management. 3 to 4 lakhs. Most of the interest we will pay, the kindly expenses we cannot do for that. Then we put our own way of thinking of things and the part of literature. Then finally we could get the time on to like the of the wheat quality function, absolutely pure and high quality DNA. Then because every time they were telling that I used to send hardly 1 or 2 milligram microgram of the conidia, a lot of food. And they grind, either it will fly with your liquid nitrogen, very difficult. But they told that I assured them that I will mass multiply at any way, I will do it. Then we mass multiply, we developed our whole the glass because it was the lockdown. We stopped our rust work in the small cabin. Then whole classes I used for the mass multiply problem. Then you can see this is the part. It has only sterile side. Everything is sterilized. Not even single, even bacterial colony you can isolate from this box. That much care we took. And seedlings, of course. You can ask me, sir, seedlings, if you have a lot of the end of it, so many things now the science is there. But for that, every 3 days or 4 days, we used to spray 70% ethanol. Starting from the germination till we inoculate. So all these sterile leaves, we inoculated only powder partners. Then we trained, very interestingly, the research associate to work with me is non-aggressive. Can you believe? One is from Madurai Kamaraj University, he completed Genomics, MSc Genomics. Another one is pure biochemistry student. So, because that was very convenient for us, because whatever you tell, they will not do their brand. So, because sometimes it's a human tendency now. Why sir is telling that you don't use the Lancet needle this way, you use only 45-degree angle. Why should not I use 60-degree angle? Because these things will come when the person is already trained. So they are not trained earlier for pathologists. So what are we told? We have executed, they executed. It was very excellent cooperation. And only because of these two guys we could get too much of media, and ultimately we got the alignment for the proof, And with that we established, we characterize and prove the 275 the isolates from the country. The IPS region, you can see that this is the region only from Lincoln and from northeast like that. And this is the paper where Dr. Angapam was telling that very recently we published from the Journal of Panchayat. Earlier it was rejected from the scientific groups, the nature group, because they were demanding that, because already one group already published in the new pathologist. With some, I should not tell us as a scientist in liberation, but the bioinformatic tools, what they use were not competent to assemble the genome, but still they published. So they were demanding that bioinformatic tools. By the time that tools were outdated, as you know, the computer science, the technologies are growing very, very fast. So, but later on, get the support from the CRP Genomics from ICR. There is a dedicated bioinformatics center. Two guys were there. We took the help. And for your information, we took almost 2.5 years to draft this. It took almost 2.5 years just to have the draft. And the Kumar and team on the other side, we were working, and any common issue, the very logical doubt only I used to clear. Plus, neither I am a biotechnologist nor the molecular biologist. I am simply plant pathologist. That's all. I don't have any background. I never worked on any molecular plant. So, but the logical conclusion I used to do, they supported me technically. And finally, we could publish. You can see this once we cultured and we multiplied and we molecular DNA. And this part, they of course, the company could care. And we could publish. And for the information, we could detect more than 126 effector genes from the past. I hope this effector genes will play a very, very crucial role in this country, not only in our country, but the global scenario. Because identifying the effector genes from the pathogen, that two pathogens like from organisms is very, very tough job. It's not easy. Now we documented. Thus, it has gone forward to 3 to 4 reviewers, very, very reputed reviewers. And the comment the reviewers itself, you have to conclude in 3 months. Each and every police what they asked was very critical because they are global leaders. And we could heard that all these are available in the public domain. So that would be made this in the free access. Anybody can access it. It's not a payment. Of course, we pay for the publication in the interest of the town users. So anybody is interested, you can go with this paper. It's very interesting work for the people who want this. I think this will give this paper will give support for the other protein, this grapes, protein, pulses, millets, so many crops we have. Of course, the government is there. So with this, let me thank the whole team. The our team, of course, everybody knows workers with me, obviously, of course, we will be classmates together here, and the remaining people, and I am very happy and to extend my thanks and acknowledgement to the Department of Plant. The whole department, I cannot mention any individual, the whole department, the whole support every time and receiving one or other day. And of course, the Ministry of Science and Technology. We are generously funded for this. And with this, I take thank you very much. And if you have any queries, we can expect. Thank you so much.
The lecture is open for discussion. So faculty and the students can interact with the methodology. It's a type of unique type of work. So those who are working in the biotropic pathogens can interact. You can get clues how to handle the pathogens and how to work with the pathogens. That will help in your thesis work or for making a publication of all. Is there any doubt like
Discovery this part.
which problem?
Yeah, when I told that there's a program that probability screening asset. That's about the IP. There we used to screen the variety. We used to say that the variety is that is score not the color is already called 0 to 9 scale. We used to give the scale is 7 or 6 like that. But we just don't mind.
That's been integrated.
Yeah, it should be integrated. Because you are giving the material for molecular reaction. You are giving the reaction. When you are giving the reaction, at least you should respect that reaction for your own material. If you ignore that, if you are interested only to push your variety, getting some publication, getting your professional career, these things, you won't work in the system. That's what it has happened. For the information, no varieties exist. Of course, some varieties are there, or they develop for the rust resistance. The LR gene, Fourier gene, they incorporated. So, because in the heat, very interestingly, the rust resistant genes are also linked to coordinate resistance. Some of them. So that is working. So when they are permitting any variety with genes, at least some these minor genes will take care of the coordinate question. And other things that except the cool climate region, we are not encountering any major outbreak like other diseases in case of. Only there is a problem in the hilly regions, northern regions, where they are going for some special type of needs for the individuals living other things. Because the resistant varieties are there, but the importance is not there from the breeders. This is the part. Compared to rust resistance, we don't do much resistance. We are not exclusively working on the wheat program. Because the reasons I told, one thing is that we don't have any established system like in case of rust. When I told in Florida, they have the very excellent facilities started from Dr. Casing and the only for rust. And only Wellington only the initiated case and we have the only.
That's interesting. So there was the exotic method of screening the missionaries. That is the also obviously a pattern. It's interesting to see the. So my question is saying we have got this.
Yeah, it's that.
Also, we have already mentioned the sugar cane. But that is the problem.
Yeah.
The problem. So whether there is any correlation between these two pathogens.
I think basically, you see, Fusarium, it's
So we have one.
yeah, it's Genera is both are same. Only the host is different. There it takes sugarcanes, here it takes wheat. But in wheat it is very dangerous, because it will put a lot of toxins.
How will this consider major issues?
Because again the same problem. You don't have a resistant source. You don't have any systematic work. If you ask any pathologist in the country, how many the races or types of Fusarium gram is the number including me? It's very flat. Because they are ignoring. You know, suppose somebody is working on leaf rust means everybody will work on the leaf rust. Any lab you see, I want to pinpoint in any particular lab. The includes starting from the students. Of course, in the IT system, we are mandated, we are compulsory to work on that crop, we are working. Otherwise, if somebody starts from some botanical, everybody will use it. Somebody starts from endophyte, everybody will work on the endophyte. Because this is the major problem that we are ignoring some part. then it will create the problem. That's what it is happening in the case of
Minor become
minor become. Otherwise, yeah, the death cap, in my opinion, it will be one of the international issue in future. Plus, it has a lot of toxins, and those I think you might have thoroughly know that some issues on the aflatoxins. Some maize grains in Rajasthan, some tribals have taken the maize with the aflatoxin presence in the problem. You go check any of the poultry feed, all are with mycotoxins. And similarly, the cycle, you test the poultry meat, mycotoxins. I think 20% of the meat, I should not discuss these things, but the first it comes from the poultry. Poultry is infested or infected meat. Lot of aspergillus means in the body. And human being know many cases of aspergillosis, disease, lungs, some cancers. So all these things are issues coming from this area of the microbiome. Similarly, the case will be in weeds. Now we have the sample map, but very difficult to because it is airborne. Sometimes seaborne. Very difficult. Probably it may have so many risks. I think you know, every time when you subculture the bacterium, it may mutate. So many issues are there to go to a plant.
Great work. Congratulations, first. I read your paper and submitted with them. And regarding this protocol, you have automation, let's say. So what is the RH inside the channel?
Genetic immunity.
Yes.
Genetic immunity means 75 to 80. So
So different ranges, first you will check, but then you optimize it and it is we have this different.
first itself, because we have the fair idea that only any field, if you go there, you feel that the climate, the complete, some discovered climate, humidity will be, at the same time, moisture also will be there. Cloudy, other things. And those kind of the weather we try to maintain insect. Because we have the gases, we have the cooling fan, and we have the habits and the controlling. It is a simple system. Only thing every time you go to regulate it, regulate and go to check it. And otherwise also, when it comes the climate is natural microclimate. Because it always cool only night. Any temperature, any rain, night it will be cool. That is guaranteed. And night it will be cool. So daytime only hardly few hours. That we overrate. That we.
So regarding this biotropic analysis between the three groups of perspective, this and this list. So is there any difference between the seasonal occurrence or days difference between the occurrence of the
Yes. We have thought that of course the whole genome maybe selfish mode. So I took our middle isolate from one native. Just spending lot of money. Why I want to sequence for somebody else days. So we have identified the whole the very regular case from one location. Otherwise, what are the race or the pathogen we collect from Shimla? They are very tiny. Then our native. So that is there, and similarly for the northwest train from the canal, it's a kind of mixed infection because you know there is a huge area cultivation, wheat cultivation in Punjab. So that might be one of the reasons that the pathogen variability is almost samples canal for facility. Otherwise, both the locations from Shimla to the Wellington, Shimla isolates are more relevant than. And after that, once we came to know that through that SRT, because all are looking in the very stringent way, and then we immediately we took care not to spread the culture. Because once any villain pathogens moves out from the communities, it will be a risk for the wheat. So that will be done.
So now but rather than you be the record plant and first time you are doing this
Mostly because we really told it's a compulsion job. Wherever you are there, you will. Yeah, that because we have, I can say, the story behind it, you won't believe. When we started, we moved to Bikaner, Rajasthan. got ARS service in Srinagar. My posting was in Srinagar. I was in sugarcane business. But that time the Excel and other issues were very easy. Every day there was some issue. So we took a voluntary transfer. So that time I took voluntary transfer from sugarcane. We said that we would together. And we asked for some other place like Mangaman. But they posted this instead of this state, they posted in that state. That will Rajasthan. That institute newly started. No building, and simple desert only. To go to inside the desert, you want to find the direction, which is north, which is east. Because in every environment, you will find the sand dunes. Like Silvan, you find that sand dunes. So, but that the institute was there, but the director, he told that there, I think in the repeat the age on food sample, where problem is the serious issue. But their food is the livelihood for the most of the research, semi. They cannot afford high price for in the economies like us. They are very, very resource poor people, but where is the major food they used to eat? So there it was the problem was the problem. The director told me, if you came from south all the way from Tamil Nadu, at least you do something for the state. That's his state.